All right, study guide B. Looking at this one, pretty much all you gotta do is get these words up here and fill it in. If you don't know anything about genetics, I would go back and watch the video from last class. But if you have a little bit of knowledge, you'll do just fine on this one. And also, I'm gonna help you. All right, so with this first one, when we're looking at this one right here, this one is gonna represent one parent's alleles. Remember, an allele is a different form of a gene. Also, whenever you have these two letters, you should think of the word genotype. So right here, put parents' alleles, and down here you could put genotype. All right, and then of course up here you have the second parent, so you can put parent two's alleles, because that word right there, alleles, and down here slash genotype. Remember, the genotype are the two genes that you have for every trait. And then right here, these four are going to represent the genotypes or the possible genotypes of the offspring. Possible genotypes of the offspring. All right, number 4. Why does each parent contribute only one allele to the offspring? I explained that in the last video. That's because you have two genes for every trait. So remember, Monday, if you paid attention Monday, you learned about meiosis. Meiosis is like mitosis, except you're making gametes. Those gametes consist of one gene for every trait that organism has. Those two gametes come together, the sperm and the egg, and then they make a new organism. So one parent is only going to contribute one half of that genetic information. So, a ratio is a comparison that tells you two or more things relate. What is a genotypic ratio? What is a phenotypic ratio? All right. So with this one, you need to know that the genotypic ratio is this. It's the probable genotypes of the offspring. A phenotypic ratio is the probable physical traits of the offspring. Now, they're not always the same. Actually, they're rarely the same. So let's just say we have big A, little a, and let's just say big A represents, I don't know, red fur, and little a represents white fur. So what we would do is say that big A was dominant for red, little a is recessive for white. So this one right here would have red fur because it has a dominant gene. And this one would also have red fur. So the phenotypic ratio would be 3 out of 4, 75% would have the dominant gene, red fur, while only 1 out of the 4, 25%, had the white fur. So the genotypic ratio is different. You have 25% that is homozygous dominant. You have 50% that's heterozygous, that's these two. And then you have 25% that's homozygous recessive. All right, and I kind of just gave you the answer for number 6. What is the genotypic ratio of the offspring in figure 5.3? Right here. The genotypic ratio, you have 1 out of 4 that's big A, 2 out of 4 that's big A, little a, and you got 1 out of 4 that's little a, little a. And the way you would write that here technically is a 1 to 2 to 1 ratio. It'd be like this: 1 to 2 to 1. That means you have 1 that's homozygous dominant, 2 that's heterozygous, and 1 that's homozygous recessive. The phenotypic ratio is like this: 3 to 1, because you have 3 that are going to have the same phenotype. So if I said big A was red fur, so if big A is red fur, this one's going to have red fur. This one. And if you have that dominant trait, you're going to have if you have the dominant gene, you're going to have the dominant trait. So this one's going to have red fur, and this one. So your phenotypic ratio is 3 to this 1 right here. This is going to have the white fur. All right, next page. What is a dihybrid cross? A dihybrid cross is whenever you have two different traits. It's a trait, it's a cross with two different traits. Why does each parent organism in the F1 generation have 4 alleles listed in 5.5? We don't have the picture of 5.5, but all you have to write is that. Hold on, let me see. 5.5, let me take a look at this image. Just write that each parent has 2 traits, which equals 4 genes. So remember that a different form of a trait is an allele. So each parent has 2 traits, which equals 4 genes. So I'll type it out. So 2 traits, which equals 4 genes. Remember, an allele is a different form of a gene. There we go. All right, so suppose the organism had the genotype big A, big B, little b. What gametes would result from this combination? Well, we're not there yet because we haven't learned about it. I can show you the answer. Let me see if I can write. Probably can't write on it with my keyboard, but the way you would figure this out, this is just good exposure. So you have big A. Oh man. Big B, little b, and then you would do a process called FOIL. FOIL works like this. First, and I know this is more complicated than you need to know right now, but you'll need to know about next week. Outside, inside, and then last. So that's the FOIL method. So this is how you find the gamete combinations for a dihybrid cross. So your 4 possible gametes would be like this. Big A, big B, remember first, and then outside, big A, little b, inside, big A, big B, and then last, which was big A, little b. So those would be your 4 gametes. You would put that outside of a 16 space Punnett square, but we're not there yet. It says, what is the phenotypic ratio that would result between 2 organisms that are heterozygous? Well, this is just good to know. This will save you a lot of work. Let's say you had a test. I would still want you to do the work, but if you had a test and they gave you 2 heterozygous individuals for a dihybrid cross, you would know that this is a special number. It's a 9 to 3 to 3 to 1 ratio. And I'll explain that next week in another video. Just know that if you're crossing 2 heterozygous individuals for a dihybrid cross, you're always going to get this ratio. All right, number 12. Probability predicts the blank number of occurrences, not the blank number of occurrences. So when we're dealing with probability, we need to know that we're going to get the average, not exact. So probability predicts the average number, not the exact. 13. To calculate the probability of 2 independent events to happen together, blank the probability of each individual event. So if we're talking about this keyword together, and we're talking about probability, that would be multiplication. Multiplication. Number 14. Figure 5.6, the probability of getting 1 coin that is heads up and 1 coin that is tails up. Well, that's pretty easy. If we have a coin and we're flipping it, and you want to know the probability, it's 1/2 each time. 1/2. All right, here's number 15. This is what breeders do. So dog breeders do this. Let's say they get a dog. He looks like a purebred dog, but you don't know. You can do the same with horses. You don't know what the true genes are. A test cross is you would take a purebred animal and you would breed it with the unknown just to see if that organism has any traits that are hidden in the genotype that are not seen in the phenotype. So, number 15, it's a cross with an organism with unknown genetics. A cross, so you cross a purebred organism with an organism with unknown genetics. All right, this law number 16, I gave that to y'all on the last recording, but the law of independent, what is independent in the law of independent assortment? It would be the genes, the alleles. The alleles separate and are inherited independently of each other. I'm running out of time. If you need to repeat, just go back. All right, here with this. Um, I really want you to try to do your best on this. I really think that if I give you a few key ones, that you'll make it work. So let's see which ones that. So this second law does not apply because genes. So I'll give you this one right here. This one is genetic linkage. What else can I give you to make it easier? I'll give you this one too, because I know I talked about it, but I think you need more practice. So this one, I want you to read it first. But Mendel's second law applies to genes that separate on chromosomes, or to genes that are so far apart from the chromosomes that they have a strong chance of being separated by a process called crossing over. Remember, that happens in prophase 1 of meiosis. Very important for genetic diversity. This is how your genes get shuffled up, so crossing over. All right, so this one right here, his first law would be the law of independent assortment. Okay. Oh, nope. Sorry. This one, I'll give you two. This one is the law of segregation right here. So erase it. I know you probably just got mad right now. Erase that. Put the law of segregation. Relax. And then right here, I want you to put the law of independent assortment. Now that's it. The rest of these you should be able to figure out. Gregor Mendel wanted to understand how traits were inherited. So he performed genetic experiments. This is for people that are still paying attention. I'm going to go ahead and give you all the answers. So he performed genetic experiments. Don't tell your peers; they probably stopped watching by now. So he crossed. So he used purebred. So he used plants that were purebred. So he examined. Go ahead and read this. Which means that the plants either had self-pollinated for so long that the offspring always looked like the parent plant. Remember, plants can pollinate themselves sometimes. He examined 7 other characteristics. First, Mendel crossed a plant displaying dominant phenotypes with the recessive. Then he allowed the first generation, so the F1 is the first generation, to self-pollinate. And then he calculated the phenotypic ratios and observed the offspring in F2. He developed the first law, that's the law of segregation. It has this law states that each parent organism has 2 copies of each discrete unit or gene, and they separate from each other during gametogenesis. And then read the rest, but you got it. All right, and if you're still paying attention per chance, let's go ahead and go over this vector vocabulary. So this one is pretty easy. All you're gonna do is write the words that go right there. You're gonna define it. So a gene It's a segment of genetic material found on the chromosome. You could also write that genes code for specific traits. An allele is a different form of the gene. And then put a word right here. So this part you have to come up with. So define these two words. If you have to go to Google and define them, that's fine. But you really don't have to. This one, there's no wrong answers as long as your definitions are right. So I'll do the first one as an example. Maybe the first two. So gene, genetic material on the chromosome that codes for a trait. An allele is a different type of the same gene. Remember that every gene is going to have different types. And in an organism, you have two alleles for every trait, two alleles for every gene. So a different form of a gene. And these two right here, you can say basically the same, but an allele is just an alternate form of a gene. All right. So phenotype, the physical characteristics. So you can write alleles. You already have the definition. You can write determine the, and then phenotype. So there is no wrong answer. All you have to do with this vector vocabulary is you have to create relationships between the words. And if you feel like that's too hard, I'm completely fine with you just defining all these words and turn it in like that. If you have any questions, email me. Y'all have a great day. See y'all when I get back.